The third kind of isomer are enantiomers. These are chemicals that form mirror images of each other. They still have the same chemical formula, but they have a different structure and completely different properties. The same elements are there in exactly the same number. It's just that the way they're attached around the central carbon is slightly different to form a mirror image of each other. These are biologically significant because only one of the forms is going to be biologically active. For amino acids, that's going to be the L form. For sugars, that's going to be the D form. This plays a huge role in pharmaceutical companies' development of drugs. If they don't have the right image or the right enantiomer, then their product is not going to be biologically active. If they can purify the active isomer from the inactive, you get a drug that's way more effective. If you have a mix of the two, as most drugs are going to be, then you have something that is not as biologically active. And so this is something that they have to consider when they are developing new pharmaceutical drugs. So functional groups. These groups are going to add properties to hydrocarbons. So remember, hydrocarbons, compounds that are strictly carbon and hydrogen, are nonpolar and hydrophobic. When we start removing these hydrogens and replacing them with different functional groups, we get a molecular structure that has a whole lot of different properties than the original. Sometimes you're going to see an R. That means where the rest of the compound is. It can be a carbon chain. It can be a carbon chain with several branches. It could be a ring structure attached to that functional group. So, as we go through the functional groups, remember, you need to know their properties, you need to know their name, and you need to be able to recognize them if you are given a compound. So as I go through these, I'm going to talk a little bit about them. Methyl groups, very important when we get to gene expression. However, these are nonpolar. They are mostly going to be found in lipids right now. We will look at them a little bit more specifically when we get to gene expression in a later course. Hydroxyl, normally an OH group. These are polar, found mostly in carbohydrates, and they are usually compounds known as alcohols. Carbonyl. This is where you have a double bonded oxygen. There are two forms of carbonyl groups. You have the ketones and the aldehydes. The one way I like to remember this is the ketones are with the O in the middle. That means the carbon bonded oxygen is on a carbon that's in the middle of the compound, not a carbon at the end of the compound. So an aldehyde is the carbonyl group where the double bonded oxygen is on a terminal carbon. A terminal carbon is just a carbon at the end of the molecule. The carboxyl group, this is a very important group. It is acidic. The hydrogen on the hydroxyl group is this guy right here. That hydrogen is going to be ionized off, dissociate in an aquatic environment, so cells, aquatic environment. This is also known as a carboxylic acid, and it's a component of amino acids. That's where the acid part of the name is coming from, the carboxylic acid. Very, very polar. And it's considered to be acidic because it donates a hydrogen ion. Whereas the amino group, the other component of an amino acid, is basic. It can form NH3 positive. It is also known as an amine. So the amino group is basic. It can take on another hydrogen ion to form NH3+. It is polar and is also known as an amine group. The sulfhydryl group is polar, slightly polar. However, it forms a covalent crosslink in proteins. Very, very critical to stabilizing protein structure. We will see that in the next video set. It is otherwise known as a thiol. And lastly, phosphates. The organic phosphate is very, very polar with 2 negative oxygens. We see this found in nucleic acids and in ATP, adenosine triphosphate. You'll get to know ATP when we talk about cellular energetics, because this compound is used for quick cellular energy, and it's how processes are going to be energized. So, are functional groups the same? Or are these two molecules the same? Carbon-based structure looks the same. Everywhere you have a point in this molecule, you have a carbon. So if we look at some of the differences, there's only a very few differences in the functional groups that are on these two molecules. Very similar structure, possibly similar function. But no, you don't want to get these two molecules confused. Why? One is estradiol, the other one is testosterone. Both of them are steroid-based hormones. They are responsible for different secondary sexual characteristics in mammals. Very similar structure, but because you have the different functional groups in a different location, it completely changes the properties. It changes the tissues that they target and what they tell that cell to do and what they tell that tissue to do. So completely different, but only the differences are in the very few functional groups that they have. So similar structure, but functional groups change the properties. And shape is related to function in biological compounds. And see you next time.
Hey guys, what you should have learned today at the end of 1.1 is 1. Water is polar due to those polar covalent bonds that it's formed, and it hydrogen bonds, which leads to the properties of water. 2. Carbon has 4 valence electrons, which allows it to form the infinite diversity of carbon molecules. 3. Isomers. They are critical for structure and function of biological molecules. And 4. The fourth thing is that functional groups. You should know that chart because you're going to have to be able to identify what properties a molecule has based on its functional groups. See you in the next unit.